Hello viewers, welcome to the course Elements of Genetics. In today's part, we shall be studying and learning about the mutation at chromosomal and molecular level. Mutations are defined as sudden or abrupt genetic changes which are heritable, and 99% mutations are lethal. In fact, in order to understand the mutation, these are changes in the nucleotide sequence of DNA. They may occur in somatic cells and are not passed to offspring, may occur in gametes, that is eggs and sperm, and be passed to offspring. Before we understand that whether mutations are helpful or harmful, let us know that mutations happen regularly. Almost all mutations are neutral. Chemicals and UV radiation cause mutations, and many mutations are repaired by enzymes. Some type of skin cancers and leukemia result from somatic mutations. Some mutations may improve an organism's survival and beneficial in nature. Before we proceed to understand type of mutation, let us appreciate and link from our background knowledge that we started from Mendelian genetics and found out that the inheritance or hereditary characters are passed through certain factors which ultimately we came to know about chromosome. The later advances in the chromosomal theory and discovery of DNA showed us that these are the genes which are responsible for the expression of phenotypic characters in the organisms. Now, mutation is a one kind of exception to the Mendel's law, that these are certain abrupt changes. There are changes which are induced at chromosomal level in meiosis or in cell division, and which we refer as recombination and crossing over. But mutation is a different kind of change which suddenly occur because of spontaneous natural region or certain artificial regions. Now, there are chromosomal mutations which may involve changing the structure of a chromosome or the loss or gain of part of a chromosome. 5 types of chromosomal mutations are known, that is deletion, inversion, translocation, non-disjunction and duplication. Deletion means due to breakage, a piece of a chromosome is lost. Here in this diagram, we see a pink colored piece which is broken away from the chromatid or chromosome, and after the reunion, the chromatid or chromosome is without that pink color piece, which is known as the deletion. Inversion means the chromosome segments break off, segment flips around backward and segment reattaches. As the diagram shows that there are different diversely colored segments on the chromatid, the pink and the green. It breaks off, that it changes its position and then reunites in the changed position on the chromosome. That is why their position is inverted or reversed, and that is why this kind causes a change in the genetic constitution of the chromosome, and that is the mutation. Duplication, as the word says, that it is the repetition, occurs when a gene sequence is repeated. Here in the diagram we see a structure of a chromosome where a segment is in yellow band, and in the side diagram we see that this yellow band is repeated and it increases in size, and therefore it changes the genetic constitution of the chromatid or chromosome, and that is why this is called a mutation caused by the duplication in the segment of the chromosome or the chromatid, thereby causing mutation. Translocation involves 2 chromosomes that are not homologous. Part of one chromosome is transferred to another chromosome. It is important to differentiate between recombination and crossing over. That recombination and crossing over takes place between homologous chromosome, and the translocation takes place between non-homologous chromosome. And the rest of the procedure is same, that a part is broken and fused back to the non-homologous chromosome, causing the change in the genetic constitution or the mutation. This is an diagrammatic explanation of translocation. There are chromosomes before translocation and there are chromosomes after translocation. The chromosomes are non-homologous in the sense that they have been referred as capital A, capital B, and the segment have been shown that there is a transfer of the chromosome, exchange of the segment of chromosome between non-homologous chromosome, that is why causing the translocation and the genetic change causing the mutation. The non-disjunction is on account of failure of chromosome to separate during meiosis. It causes gamete to have too many or too few chromosomes, and non-disjunction is mainly responsible for expression of several genetic disorders like Down syndrome, Turner syndrome and Klinefelter syndrome. The Down syndrome, 3 21st chromosomes are there. In case of Turner syndrome, there is a single X chromosome, and in Klinefelter syndrome, there is a presence of a Y chromosome with 2 X chromosomes. So, these are sex linked mutations, and the non-disjunction is caused during the process of meiosis. That here in this process of meiosis, we can see that chromosomes are separating and the non-disjunction starts from this process, and finally as an end product there are the 2 daughter cells which are having 1 chromatid less on account of non-disjunction, and 1 chromosome is more in 2 daughter cell on account of this non-disjunction, causing by severe genetic changes in the daughter cells and the mutations. These are chromosome mutation explained through animation. We can see the deletion, the inversion, the translocation as we explained earlier, and the duplication, and this is the original chromosome. Now, this situation is explained here, that duplication, deletion, inversion and another kind of inversion are shown in the chromatid by a sequence of A, B, C, D, E, F, G in the normal or the original chromosome. In duplication, we can see that there is A, B, C, duplication of D sector and F and G. In case of deletion, there is a deletion of the D sector and we can only find A, B, C, E, F, G sectors. In case of one kind of inversion, we see that inversion is in case of D, E, F sector, and in another kind of inversion, we see that there is an inversion in the case of B and C sectors shown here in this diagram. And all these duplication, deletion, inversion are responsible for sudden and abrupt genetic changes in the chromosomal constitution, and therefore, they lead to a kind of mutation. The gene mutations are the changes in the nucleotide sequence of a gene and may only involve a single nucleotide, may be due to the copying errors, chemicals, viruses, etc. Hitherto, we have come to a point where there is a second kind of mutation will occur, that is at the gene level. Till this point, we have tried to understand the change in the genetic structure at chromosomal level, that what kind of changes in the shape of chromosome will take place on account of deletion, inversion, translocation or chromosomal disjunction, but the certain kind of mutation can also take place at gene level. And we should recall that gene is the functional unit of genetics or the functional unit of inheritance. Gene is the responsible for synthesis of one functional protein or one functional polypeptide. And we also recall that gene can be in the form of a triplet code, and triplet code is nothing but a sequence of 3 nucleotides coding for one particular kind of amino acid. So, the mutation at gene level may be at the level of the large number of nucleotide or may be at the level of a single nucleotide. Again, the types of gene mutation include point mutation, substitutions, insertions, deletions and frame shift mutations. The point mutations mean change of a single nucleotide. It includes the deletion, insertion or substitution of one nucleotide in a gene. The example of point mutation has been found in case of sickle cell disease is the result of one nucleotide substitution. It occurs in the hemoglobin gene. There are genes responsible for hemoglobin, or in other words responsible for the synthesis and production of the red blood corpuscles or RBCs. And if there is a mutation or a point mutation in the gene responsible for the production of RBCs, they lead to the sickle cell disease or sickle cell anemia, which we can see in this diagram that the normal RBCs are round and oval, whereas the sickle cell RBCs are deformed in the structure as a semi-crescent or crescent-like structures are there. In case of frame shift mutation, as the name indicate, that inserting or deleting one or more nucleotide, it changes the reading frame like changing a sentence. Proteins built incorrectly. Let us recall that the genes is nothing but a sequence of nucleotide in a particular order which will work as a code, and according to the code there would be the reading by the transfer RNA through code anticodon reaction, and because amino acids are attached to the tail of the transfer RNA, there will be a particular sequence of the amino acid in a protein. So, this is a complete question of a particular arrangement. The change in one alphabet changes the entire reading sequence. How? That we have to see, and that is the frame shift mutation. This is the point mutation that changes in one or few nucleotide, and here the substitution, say for example, a sentence is being read, the fat cat ate the rat. Now, if we change a single word, it will have to be read as the fat hat ate the rat. This is one example how this sentence reading is changing, and definitely it will be having an implication on the reading of the anticode and the reading of amino acid and ultimately the protein. Second example is of insertion, that a new nucleotide is added. For example, here the fat cat ate the rat. After insertion, it would be the fat cat XLW ate the rat. So, again by inserting only one nucleotide in the entire sequence, the reading has changed. The example in case of deletion is like this. The normal sentence is again the fat cat ate the rat. After deletion, it would be read like the fat ate the rat. So, deletion has removed certain nucleotide, and there is a frame shift in the sentence, and the meaning of sentence has entirely changed. And when we say that the meaning of the sentence has entirely changed, it means the genetic implication of that gene has changed, and that is the mutation at the gene level, at one nucleotide level, or in other words the frame shift mutation. The gene mutation, we continue that frame shift mutation shifts the reading frame of the genetic message so that the protein may not be able to perform its function, as in case of the insertion and deletion we see that if we add one word, a new word H, or remove one word, the H, the implication is again on the sentence H. We can read from this diagram that the meaning of the sentence has continuously and it conspicuously changed the meaning of the sentence, which again refer to the gene mutation. Now, the significance of mutation, the most are neutral. Examples are eye color and birthmarks. Some are harmful, like sickle cell anemia and Down syndrome, and some are beneficial. Sickle cell anemia to malaria and immunity to HIV. This is a very important point to understand the genetic significance of mutation. Most are neutral means that there are visible effects of genetic mutations, but they are of no consequence. For example, the eye color changes, but there is no effect on the visibility or the seeing ability of a person. There are certain appearance of certain birthmarks, which are actually of no consequence on the health of the person. So, they are of the neutral character. Some are harmful. Certain mutations, as we just studied and tried to learn, that the hemoglobin or red blood corpuscles, red blood cells are not properly formed. In that case, the health of a person is adversely affected by production of large number of sickle cell anemia, and the blood become deficient in the hemoglobin, and that has to be cured. There are certain genetically defective babies, and various kind of syndromes are identified which are of genetic nature, out of which Down syndrome is the most popular. And this Down syndrome is because of the breakage in a particular chromosome. Some of the mutations are beneficial. For example, it has been found that the sickle cell anemia sometimes provide resistance to malaria, and sometimes it has also been found that there is a hope of getting some immunity to HIV virus, the most dreadful virus, where the fatality is almost certain. That mutation might lead to find a cause of immunity toward HIV. Now, question arises that what causes mutations? There are two ways in which DNA can become mutated. Mutations can be inherited from parent to child, and mutations can be acquired through environmental damage like ultraviolet radiation or mistakes when DNA is copied. The chromosomal mutation, the most common, as we just discussed, the Down syndrome, that chromosome 21 does not separate correctly. They have 47 chromosome instead of 46, and children with Down syndrome develop slower, may have heart and stomach illness, and vary greatly in their degree of intelligence. The certain defect in the number of fingers and number of thumbs in a child is also due to the genetic mutation at chromosomal level. And these genetic diseases are one of the most dreadful diseases caused by mutation, because there is absolutely no cure of these genetic diseases, and they are inherited from the parent to the offspring. This is the structure of a normal male, where the idiotype or the karyotype of the normal male is shown here, as the diploid number 2n number is 46, and the chromosomal mutations example, one very famous example is cry do chat. This is the deletion of material on 5th chromosome, characterized by the cat-like cry made by cry do chat babies and varied levels of mental handicaps. We observe so many kind of the mental disabilities on account of the genetic mutations. In fact, the large number of mentally retarded babies are solely owing to the genetic mutations, and these genetic mutations are being caused at the level of the chromosomals, either the deletion, inversion, translocation or disjunction, whatsoever may be the reason. But as a result, the offspring or baby suffers from a kind of the mental retardedness, physical handicapness, which is almost incurable. Only a proper care and living with and training part can help the child and the parent. The mutations related to sex chrome lead to several kind of abnormalities which we observe in society as intersex expressions, and this is referred as Klinefelter syndrome, which are caused by the abnormal behavior of the sex chromosome, that is double X with one Y, double X with pair of Y chromosome and triple X chromosome with Y chromosome, and they either lead to the male sterility, intersex behavior, small testicles and the breast enlargement, a female behaving like male, a male behaving like a female, and ultimately their intersexual nature. They are all related to the genetic mutations caused at the level of the sex chromosomes, and these are XYY syndrome, again the examples of sex chromosome abnormalities, that although there are normal male traits, but they are often tall and thin. They are associated with antisocial and behavioral problems. So, it is not only the external appearance which is sometimes governed by the chromosomal or the gene mutation, but it is the behavioral pattern also of a particular child or a particular offspring that we think that it is of psychological nature, but the deep investigation may result that this different kind of or abnormal social behavior may have some genetic origin in the mutation at the chromosome of autosomal level or at the level of the sex chromosome. The sex chromosome mutation include the triple X behavior, that is known as trisomy of the X chromosome. It occurs in the female, and obviously there is no visible difference, but certainly they can be of the tall structure, they have a limited learning ability or learning disabilities are there, and their fertility may be limited also. The gigantism is a phenomenon known on account of the chromosomal mutation, and when we observe a certain kind of abnormal behavior in case of height of a person, the phenomena is referred as gigantism, more than 6 feet tall or more than 6.5 feet tall, or it is a dwarf in the height. All these kind of the abnormality in a particular height structure is referred or is attributed to the genetic mutation at chromosomal level or the gene level. Now, what is important to understand that there are certain agents in environment as well as certain chemicals which has an ability to cause mutations. The substances which can cause mutation are called mutagens, and the mutations can be natural or artificially induced. The mutation inducing substances or agents are called mutagens, and they include mustard gas, mercurial compounds, colchicine, gamma radiation, etc. The mustard gas is a very popular mutagen. The mercurial compound, unfortunately, so many agrochemical have the mercurial compound, and they may lead to certain kind of mutation in the agronomically important crops. The colchicines are responsible for the breakage of the spindle at mitosis and meiosis level, and therefore responsible for abnormal behavior, particularly leading to the disjunction. And gamma radiation is also responsible for causing deletion or inversion at the chromosomal level and also at the level of the frameshift mutation or once nucleotide level or gene level in other words. Mutation is important in breeding and evolution. As we have seen that mutation effects can be usefully explored and exploited in plant breeding, and it helps in the improvement of the crop plant, as well as it also contributes in the process of evolution, that is the process of speciation as envisaged by Darwin. There is a role of mutation. Today we have learned about the mutation, one of the important aspects of genetics, and mutations are referred and defined as sudden and abrupt changes in the genetic constitutions which can be heritable, and mostly the mutations are heritable, and in certain cases they are non-heritable also at the somatic level. Yet the mutations, they take place either naturally on account of the environmental conditions, or they are induced by certain agents which are called mutagens. And we have seen that the chromosomal mutation and the gene mutations are the two types of mutation, and in chromosomal mutation there are deletion, inversion, translocation, and the chromosomal dislocations are the important features. And at gene level we again see that it happens at one gene level or a nucleotide level, and whether it is at chromosomal level or at gene level, it causes serious impacts and implications on the offspring in terms of their phenotypic expression. And we observed that so many genetically defective offspring is on account of the mutation, which unfortunately take place, and there is absolutely no cure with the medical science as per today. But there are certain hopes in exploitation of the mutation in crop breeding program as well as in medical science, that we find a source of malarial resistance as well as immunity to HIV in the source of the mutation. And lastly, we observed that the mutagens are those chemical agents which are responsible for causing mutation. They include mustard gas, the UV radiation, the certain chemical like colchicine, and the use of these mutations including that of gamma radiation are useful in crop improvement program and in inducing health, identifying certain strategies in medical science. Thank you. I hope you enjoyed the lecture.